Hello everyone, my name is Visalhounuo. In this video, I'll be explaining on germ tube test. Germ tube test is a screening test which is used to differentiate Candida albicans from other yeasts. Germ tube formation was first reported by Reynolds and Bird in 1956. When Candida is grown in human or sheep serum at 37 degrees Celsius for 3 hours, they form a germ tube which can be detected with a wet potassium hydroxide film as filamentous outgrowth extending from yeast cells. It is positive for Candida albicans and Candida dublinensis. Approximately 95 to 97% of Candida albicans isolated develop germ tubes when incubated in a proteinaceous media. Principle of the test is formation of germ tube is associated with increased synthesis of protein and ribonucleic acid. Germ tube solution contains tryptic soya broth and fetal bovine serum, which are essential nutrients for protein synthesis. It is lyophilized for stability. Germ tube is one of the virulence factor of Candida albicans. This is a rapid test for the presumptive identification of Candida albicans. Requirements of the test are sheep or human serum, inoculating loop, spirit lamp, cover slip, pasture pipette, microscope and incubator. Procedure of the test is: first, take about half ml of sheep or human serum in a small tube. Fetal bovine serum can also be used instead of human serum. Then, using an inoculating loop, or you can use a pasture pipette. If you are using an inoculating loop, sterilize the inoculating loop in spirit lamp, then touch a colony of yeast culture and gently emulsify it in the serum. Too large of an inoculum will inhibit germ tube formation. You should keep that in mind. And then incubate the tube at 37 degrees Celsius for about 2 to 4 hours. After that, transfer about a drop of the serum to a clean glass slide for examination. Then place a cover slip on top of it and examine under the microscope at low power and high power objectives. Coming to the observation here, under the microscope, what you will see is the yeast. If the given specimen is a germ tube formation yeast organism, then you will see the formation of germ tube under the microscope here. You can see the mother cells and then the budding cells under the microscope. Coming to the results and interpretations. If it is a positive, you will see a short hyphal extension arising laterally from a yeast cells with no constriction at the point of origin. Germ tube is half the weight of 3 to 4 times the length of the yeast cells, and there is no presence of nucleus. Example, Candida albicans and Candida dublinensis. If your test is a negative, then you will see no hyphal extension arising from the yeast cells or a short hyphal extension constriction at the point of origin. Examples are Candida tropicalis, Candida glabrata, and other yeasts. Quality control of the germ tube tests are: positive control is Candida albicans, and negative control is Candida tropicalis, or you can use Candida glabrata. Limitation of the test: Candida tropicalis may form early pseudohyphae which may be falsely interpreted as germ tubes. The yeast formerly named Candida stylatoidea also produce germ tubes. However, it has been combined with Candida albicans and no longer exists as separate species. Then, This test is only part of the overall scheme of identification of yeast. Further testing is required for definite identification.